I'm working for Bristol Myers Squibb, and if you're not familiar, Bristol Myers Squibb is a global pharmaceutical company. We are focusing mostly on, we are a leader in immuno-oncology, so a lot of my talk is touching this subject. First I want to thank the organizers for allowing me to share our work on an enterprise level NGS platform. We've done this in cooperation with our partners from Seven Bridges. A fun story, when I accepted this invitation and started working on my slides, I did not realize I have to go not through one, but through 2 legal reviews. Because, I mean, just imagine my excitement. First you have to go through a US legal team, they chew through your slides, throw some things out that they think are not relevant, and then you go to the UK legal team who goes and throws some other interesting stuff out. So anyway, there are some blank spots in my presentation, and I'll try to fill these blanks. So first of all, why BMS is interested in NGS and how it's relevant to pharmaceutical development. Well, first of all, NGS has been around for a while, and it has started to make some real impact in pharma development. And it covers pretty much everything from exploratory, in terms of target identification or mechanisms of action and so forth, through translational and clinical research. Not only that, but actually we found application for NGS in QCing in the production of our biologics. And this is a rather interesting success story I'll actually try to highlight to an extent at the end of my talk. So here just some screenshots from some interesting publications in this field. Just if you're not convinced that NGS has a place in pharma development. So it's actually, history is really good with respect to NGS. Francis Collins has this perspective on the upper right corner about the marketing authorization by FDA for an NGS machine. That's the Omnia machine, and it's obviously a tremendous thing that FDA would go through that market authorization. They saw the opportunity for an impact right there. And beyond that, really the NGS has made an impact in a number of areas, in looking at prognostic and predictive biomarkers, and we are looking also at PK/PD. You heard some of that in Mark's talk. Beyond that, there are completely novel modalities we are starting to look at to explore not only potential point mutations that can tell you something about the patient and the cancer that they've developed, but we are starting to look at the whole landscape of a cancer, things like tumor mutation burden that are starting to make an impact, and potentially a very good predictive biomarker for immuno-oncology treatment. Unfortunately, I don't have this paper here because it was thrown out, but you can Google TMB and IO treatment and you'll see what I meant to tell you. And further, we actually can look at things like circulating tumor DNA, and it's great because it's non-invasive, or at least not as invasive as a biopsy, and it can tell you more things than a biopsy can. Biopsy will tell you what's in this biopsy. It cannot tell you everything about the cancer. When you look at plasma DNA, and whether these are exosomes or circulating tumor cells, you may find mutations that are not present in a biopsy. And there are reasons for that, biological reasons, not technical. For example, you may be seeing DNA that's coming from metastasis somewhere, and it can tell you something about how to treat this patient and what can lead to a relapse or resistance to drug treatment. So there are so many questions we can ask of the data that maybe we haven't even figured out yet. So here I just, on the left side, I did a quick search, basically a word cloud for NGS and clinical. And it's obvious that people have really focused on the low-hanging fruit, which are pathogenic mutations, many of them already described, and creating panels. And this is not only in cancer, obviously it's rare disease as well, but in cancer as well. Now, on the right side is where we are moving to. This is a team at, not Memorial Sloan Kettering, I'm sorry, not MSK, this is from Mount Sinai. So this team actually looked at expression in bladder cancer, and they found that there are particular expression signatures that can predict response to IO. So that's another way of looking at the global landscape of a tumor and finding a novel modality, a completely, not just looking at individual genes, but whole signatures that can tell you, is this patient going to benefit from IO treatment? It is extremely important. We don't want to treat patients who are not going to benefit from this therapy, and vice versa. Obviously, there are people who may benefit and they may not know it. So developing those kind of novel modalities, novel cancer biomarkers is tremendously important. And in the middle, there is nothing, because that's what our legal team didn't like, but it's again related to overall survival and association with TMB. Again, something that is pretty hot in the press. There are a number of publications, high-profile publications I'm sure you can find. All right, so all these opportunities come with some challenges. And obviously, and you heard already that the word petabyte comes to mind, right? So this is where we were a couple of years ago. And this is actually BMS data. That's actually the amount of data we accumulated by 2016. This is in log10 scale. So we only started working on NGS data in sometimes 2012, 2013, and we didn't have too much efforts back then, but it started quickly exploding. And by the 2016, sometimes actually in 2015, we figured out there is no way we can scale our internal capabilities and be able to deliver data on time. So what we decided is we need a novel concept on how to process and store that data. We decided the best way is to go to the cloud. And we went to the cloud, and that solved a lot of our problems in terms of scalability. However, it's not just the size, it's also the complexity of the data. There's so many ways you can process that data. Virtually every processing step is some sort of a data reduction, and you can reduce that data in many different ways. Now, on top of that, you have some just user-centric issues, like some people like user interfaces. Some people want to touch only APIs. Computational biologists, they don't want to touch web interfaces. So we are looking for actually a whole platform that can support both web UI, API. It allows project management. It is flexible, so you can put other things into that. And on top of that, it has to be scalable. So obviously, it should be in the cloud. Cloud is great unless you have a very predictable stream of data. It should also have the ability to track cost and optimize pipelines so you can reduce cost. And last but not least, it has to fit into some sort of reproducible research framework. So this is the BMS enterprise system. To a large extent, it's based on 7Bridges platform. We've added a lot of pieces to it. So as you can see, BMS has quite large geographic footprint. We have sites in India, we have sites all over US, soon we will have sites probably, if the acquisition with Celgene goes through, we'll have sites even in Europe. And all these sites should feed through the same data processing pipelines. So this is called NGS 360. This is our kind of unifying unit. It feeds the data from all the sites and gets them to 7bridges, where the data processing actually occurs. And from 7bridges, data flows back into different, through different APIs into our warehouses. And the warehouse is really important in this platform because they allow reproducible downstream analysis. Okay, so just a little bit about the 7bridges platform. I'm not an expert on the platform, but I'm using it heavily. So from my perspective, what's important about this platform is it's based on CWL, which is an open source pipeline building platform, which is great if we decide to export our pipelines out of 7bridges. It has a lot of existing apps, and that's great because we can take these apps, use them as they are, or we can modify them like we've done with the RNA-seq pipeline that's on this graph. We've added a lot of pieces to it, so it fits our needs, especially on the QC side. That's actually extremely important because one bad sample can really throw out good results from a clinical trial, and that's disastrous. This platform also has project and group level access control. Again, very important when dealing with clinical data. It has, as I mentioned, pretty mature API. And for those of you who have ever dealt with clinical data from China, you would be aware that you simply cannot export raw patient data out of China. That never happens. You cannot do it. So for us, it's very important that 7bridges has geographic footprint in China, and our clinical trials in China, the data can go through the pipelines, the same pipelines we're using at US. So everything is standardized and reproducible. And then we can export this secondary data set to US for downstream analysis. So that's very helpful for us. And finally, something really good for in terms of cost control. We're spending a lot of money on data processing, obviously. We have thousands of clinical samples going through the pipeline every year, and we run a lot of different pipelines on these samples. So the cost is pretty high. And what we can use is something called a spot instance. And if you are not aware with the Amazon lingo, these are instances you bid on. They don't have a fixed price, but you can get them usually much cheaper than the reserved instances. And I think last year we saved something like 200K by using spot instances. All right, so I'm not going to go through this slide, don't worry. I just want to show you how complicated this is. And BMS is not a software development company. We don't want to deal with user interfaces, with APIs, with project management, all these components that you see on this graph ourselves. This would mean that we would have to hire, maybe 5-10 new people, and then we have to maintain this platform and expand it, and that's not where we want to be. That's not our core business. So we're pretty happy to partner with 7bridges. I'm sure there are other companies out there, but that we've decided to work with 7bridges, and we've been pretty happy with how things have evolved over the past few years. All right, so let me just switch gear a little bit more on the scientific side. Actually, I'm going to actually go first here. So some of the data redaction processes we're using. DNA-seq, the usual suspects, looking at TMB, I mentioned this a lot, the tumor mutation burden. We're looking at MSI, tumor purity, which is actually quite critical because if you have low tumor purity, your MSI or TMB are not accurate enough. A a lot of novel things like looking at new antigens and splicing and fusions and those kind of things. I think most people are familiar with most of these. All right, so going back a little bit to the scientific part of, and let me show you how we've improved the RNA-seq pipeline, for example. We've added a lot of QC metrics. So we're doing all the usual. We'll look at how many reads are aligned, or how many reads align to intergenic versus coding regions. Contamination level, you know, is there a lot of E. coli or something else? And sometimes it happens that just reagents are dirty or something. So we see that once in a while. But some of the new things we've looked at is actually really cool. We are looking at dynamic range, and specifically deduplicate dynamic range. And it seems that dynamic range actually correlates pretty well, not pretty well, but relatively well with ring scores, and even better with DV200. But actually it is a better predictor than ring scores or DV200 of outliers. And you do want to highlight outliers because outliers, as I mentioned, can throw you off, and perhaps the biomarkers you're trying to discover are not going to be evident, especially if you have a couple of these in a predictor group that is going to be probably devastating. These trials are not that large. I mean, actually trials are relatively large, but we don't get NGS data for all subjects. Right. So on the right side, I want to show you a really bad sample. And this is generated in open source R library, an excellent one called DupeRadar. It's really good. And what you're seeing in this density plot, basically on the y-axis, you have the level of duplication, and on the x-axis, you have the level of expression of a gene. So you expect this really dense cloud to be low and to the left, and to the right, I'm sorry, from what you're seeing. Meaning the well-expressed genes should have much lower duplication rates. Duplication rates are high for, you know, the top maybe 5–10% of all the genes. So this is a really bad sample that we should eliminate for further downstream analysis. So again, something that we've added to the RNA-seq BMS, so to speak, RNA-seq pipeline in several regions. By the way, we consider all these pre-competitive, so we are thinking about opening all these pipelines to other people to use. All right, so another QC step that we've actually implemented, this is tremendously impactful for us. We're calling it Cohort Matcher. It's based on a published open source tool called BAM-Matcher. And it's the first time actually, I've worked with some graph theory algorithms in the past, but this is the first clear application of graph theory from my perspective. I know people have used them elsewhere, but from my perspective, this is the first time. So let's say you have clinical, preclinical samples, you have a bunch of those, let's say a few hundred or a few thousand, and they go through a number of steps, significant number of steps. So the possibility of errors increases very significantly for mislabeling and swapping. And imagine if you're swapping a predictor with somebody who is not, with a responder, you're swapping a responder with non-responder. So this is disastrous, really. And it does happen. We've seen it. I think clinical trials, roughly about 5% of the samples are swapped or misidentified somehow. So this is really bad. And a couple of years ago, we decided we need to tackle this issue. And it can be actually tackled because NGS data gives you the SNPs, the germline SNPs, right? So you can do a forensic-like approach and identify if the samples are coming from the same patient. They should share the same SNPs. Ideally, all of the SNPs, germline SNPs, should be identical, right? So if you have a bunch of samples from the same patient, by definition, they should form something called a click, or a completely connected subgraph. based on the identities of these SNPs and the overlap of these SNPs. So let's say you cut somewhere around 80% overlap, and the graph that you've created based on these shared SNPs is going to fall apart, and you're going to be left with small pieces here and there. And on the right side, this is how a clean, a good sample should look like. They should be all annotated as coming from the same patient. On the left side is what happens occasionally, which means that one of the nodes in this graph is annotated as belonging to a different patient. And it actually is very, very clear. I mean, it's actually beautiful how it works. Basically, we've been able to identify all the swaps just by running this algorithm. The important piece is this graph approach scales beyond a couple of samples only per patient and cohorts of thousands of samples. We ran actually this on over 3,000 samples. Now, again, I cannot underscore how important this QC piece is for us. Right, another thing that we've done, we're not too much into the algorithm development, but back when we started working on CRISPR, we had nothing in the public domain that really satisfied our needs. So we developed this CRISPR pipeline. A person in my team, Shuning Wang, actually published this in Bioinformatics a couple of years ago. We actually transitioned this to 7Bridges platform, and it's now available. The reason we transitioned this is nobody wants to go to Git. I mean, it is in Git, and get the Python code and the R libraries and all that. You can do it. And a couple of big genomic centers have actually done that, and they're using it. And that's great. But there are a lot of labs out there that don't have the expertise or time or resources to deal with that. So we want them, go to 7Bridges platform. It's right there. It's sitting there as an app. Use it. You don't have to install anything. And it's quite useful because actually it runs in an hour or two on relatively big data set. It gives you a good idea of where the indels are happening, how much wild type you have, how much in-frame you have. So all the things you really need to make your screening a success. All right, now I'm coming to the last piece, which is a really success story that I wanted to highlight. A lot of this is actually gone thanks to the efforts of our legal team. I'm not thrashing them, they're just keeping us safe, keeping us out of trouble, but just keep in mind that this is kind of reduced slide deck. So, why are CHO cells important? I mean, that's where a lot of the biologics are produced. A lot of monoclonal antibodies, our IO treatments are produced in CHO cell lines, and other companies are doing the same. So they're really important. Imagine the drug that you have already on the market is being produced there, and you want to make sure this is the right drug. That's the same drug that you're giving to these patients, because if it's not, they're not going to benefit. It's actually really, really bad if there's something wrong going on with these cell lines. So there are a lot of steps of characterizing the product, but also characterizing the cell line itself. So a lot of regulatory agencies have a need to know if this is the same line that they approved when you started working on this drug. So obviously, you also monitor non-synonymous mutations. We do this with NGS, and we're trying to develop biomarkers for cell line stability and efficiency. But the major thing is to really know that the junction where you've integrated the plasmid that produces the antibody is intact and functioning as expected. So we've done this work with 7Bridges. Actually, 7Bridges did all of the scientific part of that. And we had a number of plasmids, 3 plasmids where we identified junctions. And if you're not familiar with CHO genome, the assembly of CHO is not great at all. So a lot of things actually are missing. And what we worked on with 7Bridges was the novel reference-free approach to identify those junctions. Not only we were successful in one of these cases, but they were confirmed by different vendors. So this was actually a really, really great thing for us. On top of that, we showed that not only we can identify those junctions, even in the absence of a reference, we can actually quantify it quite accurately. So we had a mixture of two different cell lines that have different integration sites. And they were spiked at 1%, 2.5%, I think 4% and 5%, something like that. But you see the correlation was really, really good. And so just based on the NGS data, we can first find the junctions and then say, okay, there is a contamination. This contamination is at 1% level or 5% level. And that can guide us to decide if this cell line should go forward as a production one, which is fairly important. Now, on top of that, we actually reduced the NGS approach to a digital PCR assay, which again showed pretty good accuracy. And we've been using this actually in the practice, and it's supporting a lot of our work in talking to regulatory agencies and making sure that our drug is safe and efficient as shown by the clinical trials. And with that, I'll just end, and I want to really acknowledge a bunch of key people. Joe Sotkovsky, who is leading the translational bioinformatics at Bristol Myers Squibb, Isaac Newhouse, who's my boss, all of the people here from my group, and they've been absolutely excellent. Ryan, Gohar, Ariel Sasan Sujaya, Shuning, but also the people from Severn Bridges who have helped us. Bruce, I mean, we started this with him a couple of years ago. We've had a great collaboration with Severn Bridges. It's more than just a vendor and consumer relationship, I think at this point. And the other folks from Severn Bridges who actually really helped us deliver on the CHO work. And with that, I'll take any questions.